Good morning to one and all. Welcome to Chapter 4 of Frontiers in Biological Sciences 2020, the topic being COVID-19, the current scenario. Organized by the Department of Microbiology of St. Xavier's College Autonomous, Kolkata, in collaboration with Internal Quality Assurance Cell. We will begin our webinar on a holy note with Professor Lopamutra Roy. Thank you.
सर्वेषां स्वस्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां पूर्णं भवतु सर्वेषां मङ्गलं भवतु सर्वेषां स्वस्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां पूर्णं भवतु सर्वेषां मङ्गलं भवतु सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् ॐ शान्ति शान्ति शान्तिः
Thank you, ma'am, for the beautiful and serene presentation. I would now request the head of the Department of Microbiology of St. Xavier's College Autonomous, Kolkata, Professor Dr. Shudeshna Sham Chaudhary, to inaugurate our webinar with her welcome speech.
Good morning, everyone, and a very warm welcome to all of you to Frontiers in Biological Sciences, Chapter 4, 2020, an international webinar on COVID-19, the current scenario, organized by the Department of Microbiology in association with the Internal Quality Assurance Cell, IQAC, St. Xavier's College Autonomous, Kolkata. I would like to send my sincere greetings to Reverend Father Dr. Dominic Savio S.J., Principal, St. Xavier's College, Autonomous, Kolkata, Professor Batram Da Silva, Vice Principal, Arts and Science Department, Dr. Taputi Dotto, Dean of Science, Dr. Orgho Banerjee, Dean of Arts, Dr. Parthopratim Ghosh, the IQAC Coordinator, Dr. Oyun Chandra, Member Secretary of the Academic Council, the Associate and Assistant Professors of Microbiology Department and other department of St. Xavier's College, my friends, my colleagues from other colleges, the non-teaching staffs, my dear research scholars, students, and participants. I would now like to request Father Principal to inaugurate this two-day-long international webinar with an inaugural speech. Father.
Thank you very much. A very good morning and heartfelt welcome to everyone. It's a time of global distress. We are losing lives at a lightning speed. And as I speak, more are fighting this virus. This pandemic has affected everyone in one way or the other. Our lives have gone through drastic changes. Along with all the changes that the pandemic has brought on us, changes on academic forefront has been a major one. Classes are taken online and seminars have transformed widely into webinars. The Department of Microbiology's Modern Trends in Microbiology and Frontiers in Biological Sciences are held to bring together scientific knowledge across the globe to the students. Modern Trends in Microbiology allows inter-college students to participate and give presentation on topic of their likings in the field of biology, and keynote speakers also give talk on various fields of scientific development. Frontiers in Biological Sciences has been a seminar to welcome scientists and researchers throughout the world to acquaint the students with their field of work and make way for the growth and development of students. The past chapters of this seminars have been held in the vicinity of St. Xavier's College, Kolkata, for the past few years, and this time we have not given up on it either. Our motto, Nigel Ultra, translates to nothing beyond. It is perseverance that makes us walk that extra length, and it will continue to push us further. It is in such an attempt that the Department of Microbiology of St. Xavier's College, Kolkata, along with the Internal Quality Assurance Cell, is hosting Frontiers in Biological Sciences, Chapter 4 2020, an international webinar on COVID-19, the current scenario. It will highlight on the various aspects of the novel coronavirus 19 and the current take on it by the scientific community. Our 2 days webinar will have speakers on describing the virus, its genomics, where the world stands with the finding a vaccine for the disease, how our immunity is being affected by the disease, and finally, how the pandemic is taking a psychological toll on us and how to fight it better. We have indeed lived too long while taking our basic amenities for granted. I firmly believe this is our opportunity to be better, to be aware. Today we are distant physically, but we stand in solidarity to fight the invisible but well-armed enemy, the novel coronavirus. Awareness plays a pivotal role in combating the current scenario. I urge you to take this extremely seriously and exercise precaution. I feel blessed and very fortunate to be the bearer of the message that depicts the ventures already taken up by us, food supplies and other necessities to the needy and the underprivileged. The Department of Microbiology made 1,000 liters of alcohol-based hand sanitizers for distribution amongst the underprivileged. Some changes will seem as negative on the surface, but then they are just preparing you to welcome new things in life. No matter how dreadful scenario is, try to hold on to faith. Faith is what we hope for and is directly connected to good deed and responsibility. Good deed towards yourself and towards the society at large. Be kind. No good deeds are not just restricted to donations. Being kind and empathetic towards the precarious situation others are going through can also save lives. The current situation not only challenges us financially, but also pulls down our emotional well-being with it. Once again, I will take this opportunity to welcome all staff, students, participants, and our eminent speakers. I look forward to encounter the success of this webinar by expanding our knowledge and making us more equipped to fight this pandemic. Once again, a very warm welcome and wish you all the best. May God bless you.
Thank you, Father, for your kind words. Every year, the Department of Microbiology organizes Frontiers in Biological Sciences at St. Xavier's College, Kolkata. However, this year has been an exception since the world has come to a standstill with the lockdown being imposed due to the outbreak of the deadly coronavirus. Keeping social distancing as our top priority, we welcome you all cordially to this international webinar. Since we are heavily dependent on the internet connectivity, we appeal to you to bear with us in case of any technical glitches and other unfortunate inconveniences. The response has been overwhelming this year. Near about 2,000 people from across the entire country, as well as overseas, have registered for it. We hope to get enlightened by the valuable insights about the nitty-gritties of the pandemic that our speakers will be sharing with us. We are looking forward to cover not only the clinical and physiological aspects of this disease, but also addressing the heavy emotional toll that the lockdown has taken on our mental health. We are living in dark times where the only ray of hope is a life-saving vaccine. And we are eagerly expect that the webinar will be able to throw some light on where we stand in the journey of successfully developing a human vaccine against the life-threatening virus. We are indeed very lucky and delighted to have as our speakers Dr. RANADHIR Chakraborty, currently the Professor of Biotechnology, University of North Bengal, who has always maintained a fine balance between the classical and modern biology in all his works. Dr. Anuttama Banerjee, a renowned consultant psychologist, an academic mentor, an acclaimed poet and columnist, also a guest faculty of West Bengal State University Department of Psychology. Dr. Shoshirekha Mundra from New York, a collaborative immunologist and infectious disease specialist, a medical writer for more than 2 years, also an alumni of our Department of Microbiology, St. Xavier's College, Kolkata. And finally, Dr. Chandrabali Dotto from the Oxford University Jenner Institute, where she is actively working as a quality assurance manager in the clinical biomanufacturing facility and is intricately involved in the development of the vaccine against the deadly coronavirus. We are hopeful that the webinar will turn out to be an effective and a significant one in our current scenario. Once again, thank you, Father, for your kind support. Thank you, Vice Principal, Sir. Thank you, Dean of Science, Madam. Thank you, Dean of Arts, Sir. Now, I would request Dr. Parthopratim Ghosh, the IQAC coordinator, to address the webinar with his gracious words.
Thank you, Madam. Respected Father Principal, members of the Advisory Board, Organizing Secretary and members of Organizing Committee, staff members of the Department of Microbiology, distinguished speakers, dear students, and all participants. It is a great pleasure to be part of this episode, the 4th chapter of Frontiers of Biological Science 2020, hosting the international webinar titled COVID-19, the Current Scenario. On behalf of the Internal Quality Assurance Cell of our college, I welcome everyone to this international webinar. It is my privilege to put in a few words in this context. The world came to an unexpected halt when the lockdown was imposed after the outbreak of the pandemic. Educational institutions, including schools and colleges, were also shut down, keeping in mind the health and safety of students and staff members. It has been more than 3 months now that all normal activities, including learning and education, have taken a backseat. With the gradual effort to unlock, people from all walks of life are trying to resume essential work and sustain themselves in this hour of crisis. Our lives are now revolving around the various developments of the ongoing pandemic. At the center stage of all this, we have the doctors, healthcare workers, psychologists, and scientists who are the generals in this battle against COVID-19. The world is looking forward to them for their invaluable expertise to be able to tame the virus. With this webinar, we are happy to take the initiative to connect like minds through the spread of knowledge and awareness. Frontiers in Biological Sciences 2020 gives us the opportunity to witness the distinguished speakers bring to light the various important facets of the dreadful disease COVID-19. We now eagerly await the illuminating talks from our distinguished resource persons, the speakers in this international webinar. Thank you.
Thank you so much, Dr. Ghosh, for your encouraging words. I would now request Dr. Arup Kumar Mitro, the Organizing Secretary of FIBS Chapter 4 2020, to inspire us with his words and conclude the inaugural session with a final vote of thanks. Thank you.
Thank you. Good morning to all of you. Respected Father Principal, Vice Principal, Deans of Science and Arts, the organizing committee members, the honorable speakers, the IQAC coordinator and staff members of our college. It is indeed my privilege to have this unique webinar in place, and it is dealing with a topic which is COVID-19, the current scenario. Much is yet to know, but this webinar definitely will throw some light to this particular problem which is still unsolved. And hence, to organize this seminar, we are really thankful to people who are involved in organizing this particular webinar. We really thank our resource persons, namely Dr. Ranadhir Chakraborty from University of North Bengal, Dr. Anuttama Banerjee, psychologist, Dr. Shoshirekha Mundra, who is an immunologist and infectious disease specialist, and our old student, Dr. Chandravali Datto, who is also involved in the manufacturing of the COVID vaccine, that is the Shadok vaccine in UK. And definitely we are looking forward to this particular research. I really want to thank Father Principal for his encouragement, for his support, without which this webinar would have been impossible. Vice Principal, the Deans of Science and Arts, and the IQAC coordinator for the constant support and participation, the Member Secretary of the Academic Council for his support, and always the Department of Microbiology, the staff members. We had series of meetings so that we could finalize this particular webinar. We are really indebted to a group of students of microbiology postgraduate department, namely Srijan Bhattacharya, Madhura Mondol, Maitri Sarkar, Tanishta Bishwas, and also a student of Mount Carmel, Bangalore, Charnali Chatterjee, for giving us the technical backend support, which is so important for this online seminar. And definitely, I am thankful to all the participants, all the people who have registered, and I strongly believe that this particular webinar will reach such height so that you will not be disheartened. So thank you one and all. And now with this, we conclude the inaugural session, and now the technical session will start. Thank you very much.
Once again, thank you, Professor Dr. Arup Kumar Mitro. And I would take this opportunity to thank our Reverend Father Dr. Dominic Savio SJ, Principal St. Xavier's College, Kolkata Autonomous. Our advisory committee members, Professor Bertram De Silva, Vice Principal, Departments of Arts and Science, Dr. Topati Dotto, Dean of Science, Dr. Orgho Banerjee, Dean of Arts, Dr. Partho Pratim Ghosh, IQSC Coordinator, Dr. Oyun Chandram, Member Secretary, Academic Council, and Dr. Shudeshna Sham Chaudhary, Head of the Department of Microbiology. I would also like to thank all our professors as well as the participants. We will soon move to our session 1. I would now like to request Professor Dr. Mohishita Mitra Ghosh to introduce the speaker of session 1, Dr. Ranadhir Chakraborty.
Thank you. Good morning, everybody. Respected Father Principal, Father Vice Principal, Dean of Science, Dean of Arts, other members of Microbiology Department, IQAC Coordinator, and Member Secretary, Academic Council, Dr. Ayan Chandra, and all other participants who are watching this webinar. A warm welcome to the first session of this seminar. It's my proud privilege to introduce you to the keynote speaker of the first session. Our first speaker in today's webinar is Professor Ranadhir Chakraborty. Professor Ranadhir Chakraborty currently is in the Department of Biotechnology, University of North Bengal. He started his journey as researcher in life science with his seminal work on sulfur metabolism in Acidothiobacillus feroxidans under the able guidance of late scientist Dr. Pradosh Roy in Bose Institute, Kolkata. As an environmental microbiologist, he maintained a deliberate blend of classical and modern biology. He probes some basic scientific problems like gene swapping in nature, microbial communication systems, mechanism of antimicrobial resistance with cutting-edge technology of every passing time period. He has 72 publications in reputed international journals. Professor Chakraborty has already supervised 13 PhD students and has received many research grants. He is recipient of number of awards like Best Professor Award in Biotechnology Studies in 2019. He has already initiated a study on evolution of SARS-CoV-2 and its impact on pathogenesis. It is indeed a privilege for us to have Professor Chakraborty as a speaker in this webinar. With this, I would humbly request Professor Chakraborty to deliver his lecture. Sir, please.
Honorable Principal, Reverend Dr. Dominic Savio, Honorable Principal, the members of the Advisory Committee, Organizing Secretary, member of the Organizing Committee, distinguished participant, ladies and gentlemen. I would like to thank St. Xavier's College, Autonomous, Kolkata, for inviting me, and it's a great honor, and I'm delighted to take part in the webinar on COVID-19, the current scenario, organized by the Department of Microbiology in collaboration with Internal Quality Assurance Cell, St. Xavier's College, Kolkata. With this, I want to share the screen now. Please, it is showing that the host disabled participate screen sharing, okay? Can you see the slides, participant?
No.
You cannot, so you have to enable the screen sharing.
So please share it once again.
Okay. Are you getting it? Hello? Listener, please.
Yes, sir. Yes, sir.
Yes, sir, we are seeing it.
Yes, sir.
Yeah.
Yeah.
So the topic is there. Needn't to say anything about that. So this is the slide, the initial slide I want to start my lecture with. Can you recognize the picture of this one? This is a photograph of a historic character, Pandora, right? And Pandora is a beautiful lady here, which you see that she is opening a box. And this goes back to the Greek mythology, long back, written by Hesiod. In Latin, you call it Hesiodus. And it is used now with any source of great and unexpected troubles. You all know that, Prometheus stole fire from the god. And the king of the gods, Zeus, wanted to punish Prometheus. And Prometheus and Epimetheus, these are two titans and brothers who were given the duty to create animals and human beings. As Prometheus stole fire and Zeus punished him. And he wanted to also punish the entire humanity, the human race on earth. So he created this beautiful lady, okay? This goddess, Aphrodite, it looks like the beautiful goddess to whom Ephimetheus or Epimetheus was married to. But Prometheus said that you please don't accept any gift from Zeus. But she was gifted with this Pandora box. And what she did is, driven by these human weaknesses, she nevertheless opened it and releasing the world's misfortunes and plagues. So this is the perspective that from this Pandora box, really, a novel coronavirus has escaped. And we must realize, and in the words of Principal Reverend Dominic Savio, that we must realize that in our crowded world of approximately 8 billion people, a combination of altered human behaviors, there is so much of environmental changes, but there is inadequate global public health mechanism. Now that turns this obscure animal virus into existential human threats. Second important point is, in the last millennium, the more than a millennium, we have been watching a drama which was playing out in a very slow motion. Began with the viruses of wild waterfowl that host, the viruses host had switched from the waterfowl to humans and then caused human transmission. A bird virus thereby becomes a human virus. Now there is a separate trajectory now. The coronavirus emergence takes a different trajectory, but the principles are similar. The Mid-Eastern respiratory syndrome or SARS and COVID, all these apparently have their origin in zoonotic bat viruses. Now we should have a reference to this pandemic that we are passing through. The COVID-19 pandemic can only be paralleled with another pandemic that happened 100 years back. The influenza outbreak that took place in 1918. And if you look at these, you will be finding this American soldier barrack at Kansas, from where the outbreak has taken a magnanimous, dimension. So American soldiers in 1918 pandemic And if you look at the type of, medical assistance provided at that period of time. Now, we have a lot of, Bengali audience. Now, let me recall, there's a blend of history that probably Sharat Chandra Chattopadhyay was the first Bengali novelist who has used the word quarantine. And if I look back into the time when he wrote Srikanto's second edition in the Puriched, what he writes, he says in a Poddin Bela 11টা 12টার মধ্যে জাহাজ রেঙ্গুনে পৌঁছবে, কিন্তু ভোর হতে না হতেই সমস্ত লোকেদের চোখে মুখে একটা ভয়ের চাঞ্চল্য দেখা দিল, চারদিকে এক অস্ফুট শব্দ কানে আসিতে লাগিল, কোয়ারেন্টিন। খবর নিয়ে জানিলাম কথাটা কোয়ারেন্টিন। তখন প্লেগের ভয়ে, please read, it was not plague, but it was the pandemic flu, because the time frame matches with 1918 in Burma, where a lot of Burmese army, a Burmese Indian army participated in the World War I. So this what goes the translation. The next day at 12 o'clock, the ship will reach Rangoon, but before the dawn, there was a sign of fear in the eyes of all the people. A faint sound came all around, quarantine. I learned from the news that it was quarantine. And then the Burmese government was very careful for the fear of plague. Many huts have been built around a small area with a barbed wire fence, 8 to 10 miles away from the city, in which all the passengers on the deck have been taken down indiscriminately. And after living for 10 days, they were able to make entry into the city. See, this is a picture of that Burmese army that took part in World War I. Now, if you look at this picture, the Japanese school children during 1918 pandemic, and simultaneously the war was held, it's World War I. And you look at the mask, so 100 years back, there was a usage of mask in this type of high pandemic that took place 100 years back. Now, what was the estimate of that particular influenza pandemic? Till now, that is the deadliest event. We don't know where COVID will end, but we have to our reference that there are deaths amounting to 50 million. 50 million means 1 million, 10 lakhs, means 5 crores or more dead. Ladies and gentlemen, if you look at that, what happened to India during this pandemic 1918? There were 12 million deaths in India. And all the drains and rivers were filled up with corpses of that deadly pandemic. Now, when we compare this, okay, with today's COVID-19, from December 2019 to July, just one day back, there have been 0.5 million deaths due to COVID-19. And when we look at the picture of India, the people getting affected, and when it started from the China, which dropped down to these rank of 22 in terms of coronavirus affected cases, the top of the list is the US, and in the fourth comes India, where 5,86,956 cases of COVID-19. Now, these are all the figures that those who are conscious, they know it. This is the total coronavirus cases in India, rising from February 15 to June 29. You see a sharp rise from April 15, right? And there are daily new cases in India now, which is shooting up at 20K. 20 into 1,000, 20,000 cases reported per day in India. So you can see the fluctuation here. And if you look at the active cases, it is growing up. The total death down the yellow line is also getting increased. It has crossed the limit of 15K, means 15,000 death has already occurred in India, moving towards 20K, and today is 2nd July. Look to the state of Bengal. On 13th June, this is the last report yesterday evening, which I collected. The active cases on 30th June was 5,761, but total case from Bengal reported 18,559. And when we look at the picture of the entire district level, you will be surprised that, North 24 Pargana has come up to the 2nd place after Kolkata. So it was Hooghly, it was Howrah before. Now it is North 24 Pargana, which took the 2nd place of total number of cases. And when we look at this Bengal's, daily cumulative cases, you can understand that, in the last, from 30th June, it has crossed 5,400 level. And the testing and the cumulative testing for the COVID will enhance this picture later on. So this will give some picture about the magnanimity of this particular disease. And I shall be speaking about the cunningness of this particular virus. So pandemic flu, if you have a comparison between these two, which happened 100 years back, and today, which we are passing through, the pandemic COVID-19, there is a single definition can, separate it out. The reproductive number. I will explain it vividly in the coming slide, but just for quick recap of reproductive numbers, suppose an infected person is there in a susceptible population during his or her infectious period. How many more persons are infected from that infected individual? That gives you the reproductive number. So one single carrier of COVID-19 infects how many healthy individuals in a susceptible population? So we have an idea about the pandemic flu was around 1.8. And when we look back, when we look at our picture today, the COVID-19, it is swinging between 2 and 2.5. There are reasons for why this particular reproductive number swings. But even if you think about 2.5, then also it will take 60% of the entire population of the world to get infected till you say that, you have come to a stable state. So this is, there is a equation of very simple mathematics to find out really what will be the herd immunity time. Okay, so it is estimated that between 20 and 60% of the global population will ultimately become infected with the novel coronavirus. So therefore, if we extrapolate this to Indian context, which is nearly 130 crore people are there, so you can easily calculate that 20% to 60% of Indian population needs to be, infected before we say that we are now safe. And there are differences between this flu virus and COVID-19. Flu viruses, they show, seasonal pattern, but there is no such seasonality that we observe for SARS-CoV-2. Started December, now it has crossed 2 seasons, now it is July. The same is not true for this new virus, and that means there will be a seasonal fluctuation in infection. So if you look at the worldometers and take Indian perspective, you will find the same trend. Now this is a very, very important, the point which is often talked about, which is called R0 or R0. This is what I told, that is an expected number of people that an infected person could spread in her infected period. Now, this is clear. Now, there are three interesting deductions. If this R0 is less than 1, then the outbreak will become extinct. If this R0 is greater than 1, that I said in the last slide, which is 2 to 2.5, this is leading to not only epidemic, it will lead to a pandemic, and that's what the situation is. If this R0 comes to 1, means 1 infected infects 1 more, non-infected person to get infected. If that situation comes, then the population will remain constant over time. Now, interestingly, in the pandemic of 1918, there was no epidemiological data. The countries that were involved in the World War, they were severely censoring the numbers of death. And therefore, only one country was there which was free, which was enjoying the free press. That was Spain. So a lot of deaths were getting reported from Spain, and that's why, it's a misnomer. We call it Spanish flu. But now in 2019 also, you will be finding such a trend. I not go into the details about what the big developing countries are doing about, like United States of America, Russia, and even India and in Bengal. But a spectacular study was made by a group of clinicians in Hong Kong along with the scientists, which they conducted in a single family. And this pioneer work of them, which was published in Lancet, as you see, is setting the platform of the policy. So all the policy, how we are going to handle the COVID-19 has come up from this basic epidemiological data, which shooted out from a single family. This is an excellent example of, high quality clinical study. What were the important observation of these epidemiological studies? The first is that there was an attack of 83% within the family context. That means the person who got infected actually infected 83% of other family members. This is insidious, very, very cunning virus that can infect. The infection rate is so high. The second is the clinical manifestation that took place in the family. It ranged, from mild, moderate, and severe. Third, there was a child in the family which did not show any outburst of any symptom, was happily playing around, but it was spitting, COVID-19. And the finding of this asymptomatic virus shedding, this raises the possibility of transmission of SARS-CoV from asymptomatic carriers to the others. Wonderful revelation. And the fourth revelation from this study was 2 of these young adults of that particular family, okay, they were suffering from diarrhea, which suggests that the possibility of the gastrointestinal involvement in SARS-CoV-2 infection. I will detail this because this has given a tremendous access to the understanding that how really COVID can switch from one organ to another organ, right? And if it wants to switch from one organ to another or from one animal to another animal, basically there is a requirement of a receptor. And this receptor, which was first initially detected in human epithelial airways, containing a receptor called ACE2. And that receptor is also present in many organs of human body. So if there is a possibility that there is a chance infection in other organs via blood transmission, And if there are ACE receptors there, that organs get infected by COVID. So whatever I said, we created a global human-dominated ecosystem, and that actually serving a playground for the animal viruses, which is switching its source, especially its RNA viruses are highly genetically error prone because its replication and the machinery itself, it allows lot of mutation to be recruited. So this high mutation rate allows to switch to the new host in new ecosystems. So when the bats ecosystem was destroyed by human beings, thousands and thousands of bats habitat destroyed. The viruses which were to some extent not doing any harm in bat felt that they are going to be extinct. So they switch to a most populous animal in this earth, that is the human beings. And if you look at the rate of evolution, if you take the genome of the human species, it took 8 million years to evolve by 1%. But there are many animal viruses, I can tell you, that can evolve by more than 1% in a matter of days. So it is not difficult to understand why we increasingly see the emergence of zoonotic viruses. This is a picture which you are all, aware of it. But may I draw your attention to these, spikes, which are ornate. The brown spikes is one of the area which required to be studied at full depth, because that is the basis of getting attracted to an animal host, or in this case, the human cells. And how this discovered, this particular new coronaviruses that I spoke before, that it has escaped the Pandora box. And we discovered amongst the first patient in Wuhan, and there are patients who succumb to it. If you look at this picture of this radiogram of A and B, these two patients were put to ICU in mechanical ventilation. And if you look at you cannot see through the lungs. So that's the state of the lungs that happened. The fluffy opacities covering. So when the Chinese investigators, they took out the lavage from that, of the fluid, the bronchial fluid, and look under the microscope, and obviously electron microscope, they found the existence of certain viruses. So they wanted to rule out that whether it is due to some known virus or unknown virus that came. So in order to study, you need to do it in the laboratory in cell culture. So the human epithelial airways cell culture setup was done, and the viruses were infected. And then the genome was collected, sequenced, and then it was published. And we came to know that this is a novel coronavirus. Interestingly, if you look at the onset of the symptoms from 0 day to 11th day, you will be finding here in this picture that in 5 days, you do not find something very serious that you need to be admitted to in the hospital. But on the 7th and 8th day, you feel something that you need to be hospitalized. You feel the shortness of breath. And if that intensifies, you need to be pushed to the intensive care unit. So the infective days, number 0 day to 11 days, you are in a state of transmitting this virus. And if you are not isolated, if you're not identified, actually, you will constantly spread COVID-19 to others. Now, this picture is a fantastic comparative description of fatality versus basic reproduction rate. If you look at the fatality rate, means number of case reported, diagnosed, and number of death. Means death divided by diagnosed. It's if 100 is they detected, then more than 50% they die out of when there is an Ebola virus attack. So you see MERS, you see tuberculosis, that also a bacterial disease, okay, which is above in between 10 and 20 is a fatality rate. But if you look at the COVID-19, which is given in this picture, which is below 0 to 10, as I told you, the 2 to 2.5 is the fatality rate, and the basic reproduction rate is 2.225. So within 0 to 10 is the fatality percent, means 10%, it will never reach 10% fatality. Now this is the spike, which I'm going to speak into details of it. The S denotes the spike. Now this is human cell having ACE2 receptor bound to it. And this ACE2 has some function to do, and which is a very vital function. We know what is ACE first. ACE is a angiotensin converting enzyme, which is required to increase the blood pressure. So it's a hormone that gets changed. Angiotensin 1 gets changed to angiotensin 2 with the aid of ACE. It's not ACE2. So that is a vasoconstrictor, right? So ACE is a vasoconstrictor, but ACE2, it counterbalances. So it is a dilator. So it cuts angiotensin 2 and it vasodilates. So there are regions in our human body you can think of that when bloods are pumped into our brain, you require that the pressure is maintained always in a very good way. Otherwise, there are chances of cerebral attack. So therefore, there are many organs where blood rushes: kidney, lungs, and there are many organs, I will tell you, where ACE2 expresses. And all these are very much vulnerable. Why? Because being that receptor lying on the cells will attract the virus. So this I spoke about just a minute before. The angiotensin converting enzyme is ACE, which controls the blood pressure by regulating volume of fluids in the body. And it constricts. But function of ACE2, it counterbalances ACE. And here we will be talking about ACE2, which cleaves angiotensin 1 hormone into a vasoconstricting angiotensin 2, and this ACE2 in turn cleaves. So you will be finding the location of ACE2 within the human body. As I spoke, that it starts from lungs to small intestine. And you please recollect that epidemiological study that happened in the with those Hong Kong clinicians that reported. What did they say? 2 persons were affected with diarrhea because small intestine has the cells which expresses ACE2. So it is found in cerebral cortex, hypothalamus, brainstem. So you can just imagine the type of misery it can bring if it reaches this type of these organs. Now the beauty of the SARS-CoV-2, which we call as novel coronavirus, is different from the other SARS-CoV. Why? Because the receptor to which it binds, it is hidden. So this hiding process of it actually evades the immune system. The human immune system cannot catch hold of this virus. So it then stands up like that, okay? So when it stands up, it binds. And when it lies down, it do not bind. So other SARS-CoV and this particular SARS-CoV-2, the basic difference is the spike protein remains in a standing state and also in a lying state. So when it binds, right, it has to be remaining in a standing state, and for which there must be a cut in the peptide. And that is done by another human protein, which is called furin. So very interesting thing of this SARS-CoV-2 is the site that it bears in its SARS protein domain that gets cleaved by another human protease, and which is abundantly present because these type of proteases, they digest polyproteins into smaller proteins. So why is it this fluorine cleavage important? This site is so important because other beta coronaviruses, they do not have this cleavage site. And therefore, the binding affinity of SARS-CoV-2, if you compare this with the other SARS-CoV, is very high. But initially, if you leave them without that standing position, the SARS-CoV-2 is much more infectious. So some of the most pathogenic forms of influenza, they have similar cleavage sites. Now, three important things you keep in your mind. ACE2 is a human receptor, and the SARS-CoV-2 is the spike that is going to interact. And this interaction will be done, and how the molecular details of this will make you explain why this spike, the study of the spike, is so important. And you can also imagine the velocity of discoveries coming out within this just 6 months time, revealing the entire or covering or uncovering mostly the mysteries of this SARS-CoV-2 spike. Because this SARS-CoV-2 spike is actually the target for designing vaccines, okay? And the entry is very important. So therefore, if you really think something that we need to thwart the assault of, COVID-19, you need to stop it at its entry rate, entry point. Means that you require to understand this coronavirus entry point flow, how it happens. So this picture gives you, this cartoon is an excellent cartoon, which speaks about, if you look at this A, okay, the virus producing human cells, already an infected animal cell having the viruses produced within it must escape. So when it escapes, right, it has to enter into another live cell. And here the proteases do the function. So this, I talked about the furin proteases, which is going to cleave and give a standing to the receptor binding domain so that it efficiently binds with the ACE2. And it is internalized and the viral genome is released with the aid of lysosomal proteases. Now, if you look at this picture, B, I spoke about the standing position, right? And how this stands. This stands because there is a cleave here. And this cleavage site is not present in other viruses there is a peculiarity here that there is a bridge, okay? PRRAR bridge, which is not present in any of the SARS-CoV viruses, except, SARS-CoV-2. Now, this will give you the picture that I was trying to paint with my words. Now, this is the state when it is lying down. This is the lying down state. So when it is cleaved, it is in a standing state. And when it is in a standing state, the binding to the human receptor is very, very high. And as soon as it binds, okay, there is a membrane fusion and viral entry. So B, this particular table, if you look at this table, that gives you an idea that what makes SARS-CoV-2 insidious, cunning. Why? Because it can evade immune system. So when it evades, there is, why? Because it evades in a sleeping state, that is lying down state. So if I quickly recap what I said, this RBD, or this receptor binding domain, it switches between a standing up position to a receptor binding and a lying down position for immune evasion. And moreover, to fuse the membranes, they require a proteolytically activated S1, S2 boundary, which I will speak in details, nothing to worry. And that will lead to a dramatic structural change. And the whole exposition came out in a paper that was published in PNAS. And you can see it was just published a few days back, May 26, 2020. The significance of studying spike is very important, because a key to curbing SARS-CoV-2 is to understand how it enters the cells. And this SARS-CoV-2 and the previous SARS-CoV, both they use human ACE2 as entry receptor and human proteases as entry activators. So this is the commonality, but there is also a difference that I spoke of. Now, if you look at this, this gives you a structural details of human ACE2, right? So this green part is the human ACE2, and this purple part is the part that is getting the receptor binding motif. And you find these amino acids at the contact points. Similarly, for the other SARS-CoV, if you see the human ACE is also the same receptor, but the binding points are different. This is a very, very important exposition. Now let us enter into what we did, okay? In our laboratory, or our team of collaborators who are working under my supervision is a huge team which totally mapped 3,606 genomes that has been deposited, and we mapped the entire mutation across the spine. And this gives you, this is blue to green are the transversion mutations that took place, and red to brown are the transition that took place. I will deal with details when I deal with this mutation part. Similarly, this is the point, this is what I wanted to say. This is the point of cleavage, where, this S2 is released. So there are two striking domains, that is S1 and S2, that requires to be freed due to this proteolytic dissociation. And this is giving you a picture about all the mutation across that happened in this 3,606 strains, which came out from a single COVID-19 strain from Wuhan. So key to tackling this pandemic is to understand the receptor recognition mechanism of this virus, which regulates its infectivity, pathogenesis, and the host rate. So, and this is another seminal paper which came out in 14th May 2020 in Nature, Structural Basis of Receptor Recognition. The structural features of the SARS-CoV-2 RBD or receptor binding domain that increases the ACE2 binding affinity. And I spoke about, there are presence of ACE2 even within our blood cells, blood vessels. So the interface between the blood and the blood vessel, we have endothelial cells. And endothelial cells expresses ACE2. So there is a probability, high probability of COVID-19 to infect those and create an inflammatory condition within the blood vessels. And there will be a high degree of coronary strike. This is another, just a big picture of the one that I have just shown. This is a spectacular study that was made in Zurich, Switzerland. A patient who succumbed to COVID-19. After the death of the patient, when the postmortem was done and all the organs were dissected, they found that multiple organs of that deceased person were filled up with COVID-19. And that led to a series of experiments to see that whether this is spurious data or whatever you are looking at is not a real data. So they use cell culture of different organ cells they have taken. They infected the SARS-CoV-2 and found that those cell lines were infected by the same SARS-CoV-2 that caused the COVID-19. This is a list of such, a source of cell lines upon which they worked. Now, if you look at the spike protein of SARS-CoV-2 and you compare with other SARS-CoV, you find a degree of similarity and dissimilarities. It will be more explained in the next. When we take up the entire SARS-CoV spike protein and do a phylogeny, please look here that the phylogeny of our SARS-CoV, which are all under the same group, that SARS-CoV, Taiwan, Shenzhen, Foshan, Guangzhou, France, US, all are clubbed together, and the nearest one is the BAT SL, that is the BAT ACE2 binding spike domain. So this is the spike upon which they made the phylogenetic analysis. So here you have the blue, that is this is the BAT one, this is the pangolin one, and if you take this 1273 residues of the spike of SARS-CoV-2, and if you compare it, you will be finding these differences in the different zones of the spike protein. So lineage B, again, when you do a phylogeny, you find that all beta coronavirus, that is 19 virus, COVID-19, are clubbed together, and you have pangolin just up. And above the pangolin, you have the bat. So the bat and pangolin are the nearest one to this spike family that SARS-CoV-2 bear. Here you look into the details of the RBD domain. So this is SARS-CoV-2 here. and the other SARS-CoVs are there. So that gives you the region which corresponds to the RBD. This is the slide which I would like to draw all of your attention. See, this is S1, S2 cleavage. That takes place here. RNS 600, that is in SARS-CoV-2, it is 685 and 86 is the residue number. And this is the hanging part of it, which is not present anywhere. And this is only present in SARS-CoV-2. And the red arrow gives you the S1, S2 cleavage site. And this is the S2 cleavage site. And this is the comparison between the SARS-CoV-2 protein along with the other SARS-CoVs. And when you see the protein models of SARS-CoV-2 here, and if you look at the structure of bat-rat-CoV-2, structurally, someone who is not so much aware about the structure, the protein structure, will never find a difference between these two. And this is a consensus SARS-CoV structure. So if you look at these three, SARS-CoV, SARS-CoV-2 model, and the bat, they are almost similar in topography. As I said, the expression of ACE2 receptor in human beings, if you look at this, duodenum so highly expresses ACE2. This is small intestine. And therefore, there's a high chance of diarrhea when SARS-CoV infects a human gastrointestinal tract. Besides, there are other organs which expresses ACE2. So we were also interested to find out that whether or not this particular SARS-CoV will show tropism to other animals. And here we listed certain animals which are domesticated by human beings or somehow associated with the human beings. So if we did an evolutionary history that was inferred, and even it was rooted with Danio. That means this is zebrafish AC2, which is distinctly different. But if you look at this, where a human being, the pan troglodytes, that's pangolin, all are clubbed together. So there is a big possibility of all these animals which are having a similarity in their ACE2 structure can be in future a host of this SARS-CoV-2. Now we look into the genomic part of it. Now there are two ways you can thought it's a salt, I told you. The first, the entry point. Now when it has entered, it will do its job. And there you have to protect the replication. So if you prevent the replication, you are going to check the disease. And in order to understand this, you need to understand the genome of this particular RNA virus. See, huge RNA genome, approximately 30 kilobase. Most appropriately, if we talk about the Wuhan, it is 29903. 29,903 bases. So this is the complete genome. If you look at this, 2/3 of its genome is spent in non-structural proteins. And only 1/3 of it takes part in building its structure. What structure? The spike is one of its structure. Membrane is one of its structure. Okay, so I'll come to that. But think about the non-structural protein which covers basically the 2/3 of its genome. What is that? That is responsible for production of RTC. What is RTC? Replication transcription complex. A very, very complicated way of guarding its own replication. Right? So if you look at this, this is the Wuhan's, if you, from the, it has a 5' untranslated, and you have a 3' poly A. Why poly A? The basic question is, all the learners today who are listening to this should understand that, SARS-CoV-2 virus RNA is a positively stranded RNA, which is equivalent to an mRNA. So as soon as it enters, it can translate certain proteins. So therefore, plus strand upon which the protein is translated. But if it wants to build up its own genome, then that plus strand has to convert into a negative strand. And this negative strand is going to act as a template to produce positively stranded genomes. So multiple genome copies will arise only when you have a template of negative RNA. So here lies the complexity, right? And the entire machinery, this RTC, is going to do a very complex job. A huge genome of 29,907 bases. How it does? This 2/3 is not produced as a whole. There is a frame shift, okay? So there is ORF1A and ORF1B together makes that total 16 NSPs. You know what is NSP by this time? It is non-structural proteins. 16 non-structural proteins, out of which NSP12 is the guy who's going to do the job of transcription or replication. So that is RNA-dependent RNA replicase. Interestingly, this RDRP of SARS-CoV-2 is fortified, means it is guarded by 2 important proteins. What's that? NSP7 and NSP8, which increases the processivity of this RDRP. As I told you, this is very, very, this particular polymerase is a very slippery polymerase, means it is a lot of errors. And that error, it is reduced when it is processed. You have to continuously feed the primase. And there will be several pieces of primers are going to be added and the entire genome will be transcribed. And this whole 30 kilo base genome will be produced. And you can imagine the mechanism of complexity as well as how it does. So the architecture, you need to understand the architecture of the transcriptome. Means what are the RNAs that that will be produced by this genome. Interestingly, let me just tell you, there will be production of series of subgenomic RNAs. Ladies and gentlemen, these subgenomic RNAs are not mRNAs, mind it. This subgenomic RNA will be used to produce some subgenomic mRNAs. And this mRNA is going to give you the proteins which will be required for building its own structure. Right? So the architecture of the SARS-CoV-2 transcriptome is needed to be understood at full length. Here, this picture will give you the viral genomic RNA, and you will be finding here the canonical subgenomic RNAs. See? And this subgenomic RNAs are the part of that 1/3, basically, which gives rise to S protein with spike. Then there is an ORF3A. The protein function is not well understood yet. Then E means envelope. M means membrane, likewise, okay? So these are the canonical subgenomic RNAs that are going to be transcribed by that RTC. And this is the schematic presentation of the SARS-CoV-2 genome organization and the canonical subgenomic mRNAs. Now I'm talking about the mRNAs. And when it is an mRNA which is to be expressed within a human cell, it has to have a poly A tail. So you'll be finding that in all these you are having a poly A tail. So if I quickly recapitulate what I said, the genome of SARS-CoV-2 is a last strand ssRNA with 5' cap structure and a 3' poly A tail. The genomic RNA will be used as a template to directly translate the polyprotein, as you have seen that 1A and 1B, which encodes the non-structural proteins. I spoke in details, to form replication transcription complex, and which will be put inside a double membrane vesicle. Subsequent to that, there will be a nested set of subgenomic RNAs, which will be synthesized by this RTC in a manner of discontinuous transcription. And then this negative strand sgRNAs will serve as a template for the production of subgenomic mRNAs. Approximately 2/3 of the whole genome, it encodes 16 NSPs, which is somehow involved in the preparation of this RTC. So there is a -1 frameshift between 1A and 1B, leading to 2 polypeptides. And these polypeptides, now they will be cut by certain proteases in order to produce those specific NSPs. Now this will give you a quick look at what these NSPs do. You talk about NSP1, the function is cellular mRNA degradation. And it inhibits IFN signaling. NSP2, unknown. NSP3, this is the polypeptides cleaving. It blocks the host innate immune response. NSP3 is very important. It promotes cytokine expression. NSP4 is that DMV, that vesicles produced. NSP5 is the polypeptide cleaving, and also it inhibits the IFN signaling. NSP6, it restricts autophagosome expansion. NSP7, these are the 2 very important cofactors. It is a cofactor which binds with NSP12, which is a primer-dependent RDRP, as I talked in a little bit details. Likewise, you have a 16 NSPs. Some we know about the function till date. Some we are yet to find out in details the function. I skipped this because this is just an extension of that one. Now, when we take the entire genome of SARS-CoV and all related SARS genomes and do a phylogeny of the complete genome, where you see ours, this CoV2, this is here. This is here. It is a separate clan as such. So this is a polyphyletic way of evolution that took place. And the nearest one means its nearest relative is the bat one. So this has polyphyletically differentiated. So phyletically it is different from that of this SARS-CoV, the bat SARS-CoV. Now, when we look at this same picture with the common cold coronaviruses, this phylogeny in forms of the leaf, you'll be finding that SARS-CoV-2 and SARS-CoV, these 2 are the nearest relatives under this green leaf. This gives you an overall picture of what coronavirus after the entry and the exit is given here. So these are all dealt in details. That is how you translate. So as soon as it enters, I said it is exactly as the mRNA. So it is a positively stranded RNA virus, translates, and then other proteins are produced by these replication transcription complex. Genomic and subgenomic RNA is produced, and finally it is packaged, and then exocytosis takes place, and then it goes out to infect some more cells. This is what the RNA-dependent RNA polymerase looks like. So this is the RDRP, has peculiar domains, just fingers, palm, and thumb, upon which very many work people are doing who are interested in finding out drugs that can inhibit, right, the RDRP functions. And one of the promising drugs is remdesivir. Why it is so promising? Because it is a base analog. It terminates replication. So if it binds with RDRP, okay, it binds means it is an analog. While it is transcribing, this analog, this base analog, where it goes and get, you know, uplifted, immediately the translation stops. The transcription stops. And even it does not allow, the remdesivir has another important function, that is the processivity done by the other two proteins also are blocked. So therefore it's a promise that, you know, remdesivir like drug is going to inhibit the RDRP functions. As I said, you know, if you look into the details of this, this is the structure of RDRP. And when it is crystallized, it was found that NSP8 and NSP7 are both these cofactors. They make a complex with RDRP. And therefore, you always get an association of NSP8 and NSP7. So therefore, it is my urge to the scientific community also that do look at this NSP8 and NSP8 function, because NSP7 and 8 helps in doing, you know, the restricting lot of functions. It helps in RDRP process. So if you can block that, then the whole replication process will be erroneous. So this is an extension of that. Now, this is a very important area of my interest, because I wanted to look into what's happening within the genome of SARS-CoV-2. Wuhan strain released in December, and June 2020, you know, more than 3,606 genome sequences have been deposited. So when you have that opportunity and study, so we did not have the opportunity to study this 3,606 when we started working into this particular aspect. We posed some questions after we analyzed the transition and transversions. So basically, a simple geneticist is understood that, you know, there can be only 4 transitions, 4 different transitions can occur. It is theoretically possible, maximum, and transversion, 8 types of transversion. So therefore, transversion rate should be higher, but it did not happen. Transitions are much predominant than transversion. And when we looked into the details about the transition, we found that C to U transversions are predominant and overwhelming. What's the reason behind this? The reason is, you know, spontaneous deamination of cytosine. Interestingly, for any DNA, viruses or DNA, it can be corrected by error processing mechanism within the cell. But when it is an RNA, right, and when the C is converted to U, it is permanently there. This is no question of getting repaired. So these are the sites. This C means there are certain dipyrimidine sites within the genome which are susceptible to these type of damages. What damage? Means C converting to U. And interestingly, when we studied all this genome, we found an overwhelming presence of C2. I will come into the details of that. So therefore, for the students, these are the transitions. There are the 4 different transitions can occur. Pyrimidine to pyrimidine means C2U. You think that T is nothing but U, and this T is U. So these are the 4 different transitions that can take place. And transversion means pyrimidine to purine and purine to pyrimidine. So these 8 different combinations of transversion can take place. Interestingly, again, a question is answered from the basic genetics, right? That if you look at the coding, the generic code, the 3rd base is always a purine or a pyrimidine. So therefore, if there is a change in the purine to another purine, there will be no amino acid or lesser degree of amino acid substitution. So basically, these are synonymous. Most of the transition mutation must be synonymous mutation, means silent. There will be mutation, but there will be no replacement in the amino acid, right? But interestingly, when we studied the entire transitions of SARS-CoV spike, you'll be amazed, ladies and gentlemen. This transition has caused a lot of amino acid substitutions. That makes the whole thing more strange, more queer, more difficult for the scientists. We started our study with just doing, right? A simple blast of the 13 strains reported from different countries. And we wanted to find the significant alignments. 100 significant alignments out of each of these sequences leads to 1200 sequences. 12 into 100 or 13 into 100, 1300. And this 1300 sequences, when you just study the blast, hey, interesting, that it never crossed an identity barrier above 99.93. So all these genomes, they were restricted to 99.93 at best, because most of them were restricted to 99.98% of identity. That led us to go into a little bit more details. So when all these, okay, pan genomes, means 13 SARS-CoV-2 along with the reference genome, that's the Wuhan strain we did, and we found the nature of the mutations. So this is what you will be finding: the C2U, 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 C2U maximally open. Okay, so this is the mapping that gives you. So we wanted to validate this. How did we do that? By that time, you know, the 46 Indian strains were deposited. So we did the same thing with these 46 Indian strains. And again, we took some random 10 different strains of 10 different countries, and we did the same. And now we placed the mutation, right, gene-wise, 16 NSP genes and 6 and other structural genes that I talked of. We found this is plus means there's a presence of mutation. But when we did this, we found strikingly 7, 8, and 9 of all the sequences that we studied were devoid of any mutation. Lovely. So we wanted to do this with the entire 3,606 transition. Now we are having a better grip upon this mutation biology of this SARS-CoV-2. So if you look at this, A to G, G to A, C to U, and U to C, how many mutation in what NSPs and other all were mapped. And strikingly, 107 C2U mutation is located in gene S. What is gene S? It is a spike protein. So therefore, the spike protein is a direct hit of all these transitions, and C2U transition taking place at a maximum rate here and NSP3. The significance of which we are working on. Okay. So when you look at the genome-wide spectra of transversion, which is much, much less. So if I move to the previous slide, and if I talk about, the total number of mutation in 3,606 transition, strains or quasi-species, we end up with 1,813 mutations. What mutations? Transition mutations. Now, if you look at the transversion mutations, there is very less amount of transversion mutation throughout the genome, right? And interestingly, when you add up all these across the total genome about the transversion, it comes down to 660. So almost 1/3 of transition. So now we have a big picture, So this is a big data upon which we are now working upon, right? Please recapitulate this. This is that one which I showed about the S1, S2 cleavage site. And now, with all these data in our hand, now we started plotting it across the SARS-CoV genome. That what are the changes that took place all across? Now, this is a complete mapping has been done, upon which now we are selecting certain mutation, expecting that certain mutation in the spike will enhance the binding affinity. So therefore, there are possibilities that you will get much more infectious SARS-CoV-2. So which are the mutation that will lead to the enhancement of the binding affinity with ACE2? This one, just for IPR reason, I'm not going to disclose what mutation we are now working on. But this picture will give you a clear idea that the green color is the mutated spike and the cyan is the Wuhan. So a mutation in the spike have characteristically disrupted the entire structure to certain extent that will lead to the enhancement of binding affinity. Next, you will see we have plotted those contact points side by side with the mutated along with the one of the Wuhan. The cyan are the Wuhan and the green are the points. Basically, all the contact points remain the same, but the position of the different important amino acids at the ACE2 binding sites will give us more detail about all the molecular simulation data will speak about that, and that we need to experiment that what really happens if such mutation happens. Recollection, that is these points, see, F, L, Q, N, these are all amino acid position wise. And if I move back, fantastic, 501, 493, 494. So absolutely the working that we are moving in a direction which is corroborating with the data, the crystal data. We also tried, the whole phylogeny of these 13, which I spoke about. the initial 13 genome to find out whether there is what's the relationship, phylogenetic relationship between. If you look at this strain, this is the first Wuhan strain, and that diverged. The evolution took place in this direction. So we have another Chinese strain and Indian, which is making a cluster. So this is giving you a picture of understanding that South Korea and Brazil, they are nearer to each other. And Brazil, South Korea, Italy, and Sweden under the same crystal, having a common ancestor here, right? So within these 6 months, there has been a tremendous rate of evolution. And we really wanted to understand that, you see, the error replication rate of RDRP is not actually defining that how many C2U conversion can take place. So therefore, we looked into certain aspect of human aspect, that is, are we accelerating the rate of C2U? Because in all sorts of disinfection, even the airplanes, the buses, they are disinfected with UV, right? And UV leads to spontaneous cytosine to U conversion. So this deamination takes place. And for doing lot of disinfecting your surfaces, entire Europe uses, they use metabisulfite and sodium bisulfite that is being used as disinfectant, oxidant, antioxidant, and preservative agent that can also give a direction of change to cytosine to uracil even before it enters into the human body. Similar is the case, the next most prevalent transition that we have noticed is A to G. Now, how can that take place? Very interestingly, formic acid medium, the water and formic acid, which is also used as cleaner formulation, can help in directing the chemical reactions to adenine to hypoxanthine to xanthine to guanine. So A2G can also be accelerated out. So when it is outside your body on the surfaces, when you are cleaning, there are possibilities. You cannot rule out that formic acid and water can also spontaneously change adenine to guanine bases. This is my entire team which is still working on this issue. We are doing all the comparative genomics. More collaborators are entering into the whole team. I end my lecture here. Namaskar to all of you who have listened to this with full patience. Thank you very much. Hello?
Hello.
Yeah.
Yes. Thank you, Dr. Ranadhir Chakraborty, for your very informative and insightful presentation. Now, I would like to request our Professor Dr. Joydeep Ghosh to conduct the question and answer session, as well as present the vote of thanks to our speaker.
So, thank you very much, Professor Ranadhir Chakraborty, for this wonderful lecture. Now, we will move on to the question answer session. There are quite a lot of questions, but since we have some time constraint, we have selected a few for your answer at this moment, but we will send the other questions to you. When you have time, you can send us the answers and we will send to the particular persons. So the first question will be, is there any chance of silencing the production of NSP7 and other NSP proteins?
Well, if you want to silence it, you have to stop the replication part. First, the translation of the genome, as soon as it enters, you have to silence them. And for which we need to have certain drugs that is going to act on the RTC. So if the RTC can be malfunctioned, you can stop the production.
Yes.
And there are hopes, people are trying that. Okay.
Yeah, okay. Thank you very much, sir. So the next one will be, what are the chances of reinfection of a person with a different strain of the virus after recovery from previous infection?
It's a very good question, I tell you. Yes, there is a possibility of getting reinfected. And this is proved that, you know, people who have been affected is getting reinfected. Even in China, there are cases reported of reinfection. Because of its insidious nature, because of its high degree of mutability, right? So it can occur that, you know, some other strains, which is having much more efficiency to infect, is going to evade the immune system of the infected person. Okay? So it can happen that you require another set of antibody to reappear to counter those newer viral infections. There is a possibility. So we cannot say really that even after the discovery of the vaccines, how far the vaccine will remain stable is also a big question. Yeah,
Yes.
there is a question.
Yes, sir. So there is a related question that you probably have already answered, that as coronavirus has different strains,
Yeah.
is it possible to develop a proper vaccine at all?
Well, people are not giving their hopes. For example, in India, Bharat Biotech, they are doing it with the entire virus. Okay, so if that works, because it is coming to the clinical phase, and that's the best way to try first time, rather than look at the targets by looking at the spike protein. I know the spike is there. There could be several diversions of the spike. So taking them together and using that dead virus as a inoculant, that might help, and that's a promise we look forward to.
Okay, sir. So then how efficient is plasma therapy, and is there any chance of severe side effects from that?
That I'm not the proper person to answer, because
Yeah.
side reactions that some other the sorry, I cannot answer this question, but plasma therapy people must try, because you know, when you have nothing to do, you have to try all these things.
Okay, sir.
But the best thing is you wait for the vaccines.
Yes,
and already
yes.
it is in the clinical trial. But
Yes.
before that, even to a person who is ailing so much, you need to you need to look at these patients. At that time, if you have the plasma, and if you do this plasma therapy, and if that enables that patient to recover, that will be fine.
Right.
That will be fine.
Right. Right. So, according to your opinion, when will be the peak of infection be reached in India?
Well, first of all, there is a dearth of data, right? Data means we are getting the data of the persons who are being tested, found to be carrying COVID-19. There are 100,000 people, because you see 130 crore country. At least if you want to cover 10% testing, that's a huge affair. So if you don't have that data, it is very difficult to predict. But one thing we can predict for sure. What's that? When will the herd immunity appear?
That was the next question, actually.
Yeah, the next question, right. Thank you very much for putting up this question. See, the herd immunity, all of you who are hearing me, please note there is a very simple equation. What is that? 1 minus 1 by R0 into 100. What is R0 that you have understood? That is R0. Okay. So if I take 2.5 is the reproductive number. So 1 minus 1 by 1, 2.5 is equals to 0.6 into 100 means 60% of Indian population, unless gets affected, we are not seeing any direction towards herd immunity. Again, I repeat, 1 minus 1 by R0 into 100. R0 is that number, and this number is varying. It will stay somewhere else in between 2 and 2.5. So if you put that, it comes to near about 60%. And if everything does well, and Indian population, the rate of death is low, you can expect 30%, 30 to 60% of the entire population needs to be infected to get a herd immune system. And now you know how many of them. So it will take a long time to reach them. Yeah. Next question, please.
So far, you have already answered 5 to 6 major questions. The others are sort of repetitions of this, and the rest of the questions will be mailed to you, sir.
Okay, thank you.
To conclude this session, I must say that we have heard this wonderful presentation, and we have now an excellent molecular picture of SARS-CoV-2, and we have learned that the unique spike protein of SARS-CoV-2 is getting mutated, and some of these mutant proteins are probably much more efficient in interacting with the human receptor ACE2. This genomic plasticity results in the emergence of new forms of SARS-CoV-2 that are probably more infectious and hence have a greater potency of transmission. At the same time, has the potential of being more resistant to therapeutics. These data provided by Professor Chakraborty will be immensely helpful for the formulations of preventive measures as well as cure of the disease. Moreover, this study will allow us to predict the future evolution of SARS-CoV-2 and thereby guide us to design strategies to fight this and also other related viruses that might threaten human lives in the days to come. Thanks a lot, Professor Ranadhir Chakraborty, for sharing this extremely important and insightful work with us. So now over to Moitrey.
And also, thank you, Dr. Ghosh, for summarizing it so well.
Thank you, sir.
Thank you very much. Thank you very much.
I would like to once again thank Dr. Mohashita Mitra Ghosh, Dr. Ghosh, and as well as Dr. Ranadhir Chakraborty. This marks the end of our first technical session. I hope to see you in the second technical session at 6:30 p.m. And to my dear participants, there are no feedback forms for this technical session. You have to stay tuned because they might arrive at any point of time. Thank you, everyone. Until then, please take care.